Hi budding biologists, and welcome back. In this series we're going to be looking at revision for module 3. This first clip we're going to be doing a big picture mind map of all the content that we've covered so far in module 3. So let's get into it. Module 3 has been all about biological diversity. We've had 4 main inquiry questions and we're going to work through them one by one. First, we're going to look at environmental pressures that cause change, and we looked at selection pressures such as abiotic and biotic factors. Abiotic factors include things like air temperature, water availability. Biotic factors were things like competition within species. We looked at symbiotic relationships, predation, parasitism, and disease. And we looked at competition was for things like food and habitat and available mates. From there, we looked at selection pressures can be both positive and negative. Positive means that it increases the survival. For example, an increase in food means that the population would ultimately increase, whereas a negative selection pressure decreases an individual's possibility of survival. For example, increasing water temperature in the ocean causes mass death in corals. Next, we looked at a case study of the cane toad. The cane toads were introduced into Australia to eat the cane beetle. Ironically, they don't actually eat the cane beetle, but since their introduction, they quickly evolved. As they spread across Australia, heading particularly south and north, the individuals with the longer legs were selected for at the edges of the population. This is because those individuals are able to travel further, and that meant that they were breeding with other individuals with longer legs, so the genes for longer legs were selected for. They had a range of negative impacts on the ecosystem due to their toxicity. There's no known predator, and that causes competition within the native species for the same ecological niche. Another little side note while we're here, in the Northern Territory, the red belly black snakes actually have evolved to have smaller jaws. And that's thought to be because of the selection pressure of the cane toads. The ones who didn't eat the cane toads and die had smaller jaws because they were unable to swallow them. And so it's had an impact even on the native species within Australia. All right. So that's the end of inquiry question 1, looking at environmental pressures that cause change. Next, we're going to be looking at adaptations that increase survival. So we looked at the 3 main types of adaptations. The first type was structural, and structural involves a physical feature of an organism. For example, things like waxy leaves on a eucalypt, where physiological adaptations are chemical changes that occur within the organism, for example, dropping leaves or metabolic processes. The last type is behavioural, and they're things that the organism does, for example, releasing seeds after a bushfire for the Banksia, or being nocturnal for a mammal. In this part of the module, we also reviewed the theory for evolution by natural selection, and we looked at the major steps for that natural selection. So first off, we've got to have variation within the genes of the population, and that's expressed as phenotypes, which make an organism more suitable for survival. Next, there needs to be a selection pressure that's applied to the population. So selection pressures relate closely to either abiotic and biotic features within the environment. Then the most suitable phenotype or individuals within the population survive, and they reach sexual maturity and are able to pass on their genes through reproduction to their offspring. Darwin took a trip on the HMS Beagle, a world tour looking at different organisms around the world, and from his observations he proposed the theory of natural selection as the basis for evolution. Some of his research involved those Galapagos Island finches that all had the same common ancestor finch, but when they went to the different islands, they experienced different selection pressures, and this resulted in divergent evolution, resulting in new species that were no longer able to interbreed due to adapting to the different selection pressures. Another example that he used was Australian flora and fauna, that they came from different ancestors but had similar ecological niches to those organisms that are in the northern hemisphere. And those similar ecological niches resulted in similar adaptations to those environments. For example, the platypus and the water rat both have slimline bodies covered in fur, allowing them to stay warm. The last thing I wanted to review requiring this inquiry question was variation. So we need to recall that variation can either occur through sexual reproduction, where you have the mixing of genes from two parents resulting in the random expression of genes, dependent on dominance, etc. And then we also get variation in the population through mutations. Our next inquiry question was looking at how biodiversity and evolution are related. Starting off this module, we looked at the diversification of life on Earth. There's a long history with lots of details. I recommend you go and have a look at that in more detail from your notes. But generally, the diversification of life on Earth has had periods of explosion and extinction due to a range of selection pressures, and a lot of that resulted from changes in abiotic and biotic features interacting with one another. Micro and macro evolution. So microevolution is founded in the small changes over short periods of time, relatively short periods of time, and an example of that would be Darwin's finches, and they're an example of adaptive radiation, where a common ancestor resulted in a large number of different species. Where macroevolution is large-scale changes, so whole new species or whole new features within the species developing over a geological timescale. It can be due to the accumulation of microevolutionary changes as well. From there, we looked at speciation. Speciation is when there's the formation of a new species. There's two main types of speciation. There's allopatric and sympatric. Recall that allopatric means when there is a physical barrier where there produces reproductive isolation. Physical barriers can include islands and mountains, whereas sympatric speciation occurs within a species, and that's where an organism may find a new ecological niche, or there's a selection pressure within the population that results in the new species. The two case studies that we looked at was the platypus and the horse. Next we looked at divergent and convergent evolution. Recall that divergent evolution is where each species, each individual within the population experience different selection pressures. Due to the variation in the genes, there's an increase in diversity as they survive to the different selection pressures. So one species becomes two different species in divergent evolution. Where convergent evolution is where similar selection pressures are applied to different organisms of different species and they result in similar adaptations to their ecological niche. So remember, divergent is moving apart, where convergent is moving together. The last little portion around this inquiry question was we looked at the difference between the theory of punctuated equilibrium and gradualism. Punctuated equilibrium was proposed in 1972 by Eldridge and Gould and is supported by the fossil record. It states that there was periods of rapid change, biodiversity boosts, by periods of relative stability. This is in direct opposition with Darwin's gradualism, which proposes slow microevolutionary changes in a uniformed way. This brings us to our last inquiry question, which was looking at the evidence that supports the theory of evolution. The five main parts of evolutionary evidence that we looked at in this inquiry question was biochemical evidence, comparative anatomy, embryology, and biogeography. Biochemical evidence can include amino acid sequencing, where comparing proteins between species is used to determine relatedness. DNA hybridization, where the direct comparison between two species and related alleles are compared to determine relatedness. And DNA sequencing, so actually the code for the DNA being used. It provides strong evidence to show relatedness between different species to relate to common ancestry. Next is comparative anatomy. The most common example that we've looked at is the pentadactyl limb, and it's evidence for divergent evolution. It's showing the common structure is seen between a range of different mammals, that they would have had a common ancestor with that structure. The other bit of comparative analogy that we looked at was analogous structures, where there's similar structures but they're actually from different common ancestor. So for example, wings on butterflies and birds, having similar structure doesn't always mean that they're related. The last bit is vestigial structures, which are structures that are no longer used by the common species, the most recent species. For example, the coccyx in the human, that's the tailbone. Comparative anatomy provides relatively strong evidence to support the theory of evolution. However, the biochemical evidence is far stronger. The next piece of evidence that we're going to look at is relatively weak, and that's embryology. It was developed in the 1800s, and it was based on a series of diagrams that were interpretive of the developmental stages of different species to show similarities. The next piece of evidence that we looked at was biogeography, and it's looking at how separations between different countries and migration patterns results in new species. For example, Australia and New Zealand sharing similar species compared to that of things in the northern hemisphere. The next piece of evidence that we looked at was the fossil record. The first thing I'm going to review is relative and absolute dating. So relative dating is using stratigraphy, and stratigraphy is using the relative layers where the fossils are located and comparing it to fossils either side to determine the age and place in the record. This method is somewhat reliable, providing that you have enough fossils either side of the sample. The next is absolute dating, and that includes things like carbon dating, fission track dating, potassium-argon dating, and it is quite reliable. And it relies on the half-life being measured within the sample to determine its age. Fossil records have also been used to demonstrate evolution as it shows transitional forms. We looked at some examples of those in your notes. But the ultimate weakness of the fossil record is that it is quite incomplete. Next, we looked at some examples of modern evolutionary change. We looked again back at that cane toad case study and how they've changed Australia. But the new case study that we introduced at this point was antibiotic-resistant bacteria. And we looked at how natural selection resulted in antibiotic-resistant bacteria. So as we know, bacteria can reproduce asexually, but they can also reproduce sexually, and that results in the variation in the species when exposed to drugs and the fittest being able to pass their genes on to their offspring, and that means that the population over time becomes drug resistant. So that's our whole concept map for biological diversity. I hope that was helpful with your revision. See you next time, budding biologists.